Oral cancer prevention advances with a translational trial of green tea. This perspective on Cao et al., beginning on page 931 in this issue of the journal, discusses green tea extract, which was shown for the first time to have dose-dependent effects in a clinical chemopreventive setting, oral premalignant lesions. This translational trial provides important data on angiogenesis and other biomarkers on which to base future clinical research, which should include trials of green tea extract or polyphenols combined with other natural or synthetic compounds to enhance chemopreventive effects. Green tea polyphenols can also induce cell cycle arrest or apoptosis by activating p53 in its multiple target genes. It is important to emphasize that conducting biomarker evaluations within a clinical trial is very difficult and challenging, requiring, for example, tremendous effort in performing multiple biopsies and biomarker analyses in the Cao et al. Study. Undertaking clinical chemoprevention trials that incorporate biomarkers, however, is the only way to ascertain whether or not a chemopreventive agent is working at the molecular level and to understand mechanisms of response and resistance. Such biomarker studies will be critical in clinical trials of natural compounds because these agents are relatively non-toxic and thus may not be well suited to the traditional phase 1 therapy end points of maximum tolerated dose and dose-limiting toxicity. Determining biological activity by molecular marker modulation will be an excellent guide to the best methods in chemoprevention settings for these agents. Therefore, Cao et al. Are to be congratulated for their enormous effort in incorporating biomarkers into their clinical trial of GTE in patients with oral premalignant lesions, and for their contribution to the development of chemoprevention strategies using natural compounds with the potential to reduce cancer risk. Combined agent approaches are of emerging importance to cancer chemoprevention, and it will be particularly important to examine tea product-based combinations, for example, EGCG, with the potential to maximize chemopreventive efficacy, versus single agents, in part, by suppressing the emergence of resistant clones, a typical obstacle to long-term chemoprevention. Indeed, our recent preclinical study of EGCG combined with an EGFR inhibitor showed promising effects, including NF-kappa B suppression, in head and neck carcinogenesis.